Okay, then good morning also from my side. Today we start with a talk of Max Pierobon. You know him already well. He contributed with a lot of nice and interesting questions. And he was yesterday also at the panel. And you already noticed that he is one of the people who push molecular communication a lot. Somehow I would call him a little bit someone of the founder. Also, he also gives a lot of thanks to us. We have to thank him to push this field a lot and working there a lot. Also, Yaning was visiting him. She also enjoyed it a lot and learned a lot last year. So thank you for that. And I'm very curious in your talk today, and it's on the usefulness and subjective of life supporting information. So, Max, the stage is yours.
Great. So, well, thank you, Christian, for having me here. It's been a pleasure to meet each one of you in these days and to see your presentations. So today is my turn. I was thinking about what to present here on with respect to all the research that I'm doing currently, and then I saw the title and post Shannon. I got inspired in presenting some of the work that we did in information theory. And so that's why this presentation will actually go more over that work. And we'll see some of the results that we had. Some of them are actually pretty timely in the field. First of all, I want to start with a couple of slides that I used to provoke thought. They are about communication and about communication, the difference between communication in inorganic systems like the internet and all the devices that we engineer through the years until now, and the communication in instead biochemistry. So we all agree that information is a difference which makes a difference. Do you know who said that? So let me give you a hint. Gregory Bateson. He's one of the fathers of cybernetics. And indeed, when we think about what is information, which a little bit escapes, a definition, or was escaping definition until Shannon, right? And so the difference that makes a difference, it was Shannon captured with the entropy as a measure. But you all agree with me that whenever we are using the internet, for example, to write an email or a message, internet does not really care about what is written in that email or in that message, but our devices care about transmitting the information in the fastest way possible or with the lowest error possible, or both of the two. What we have here is a system that is used to transfer information, but does not really care about what we identify with semantics, right? What is inside the message is only a syntactic system that cares about transmitting information from one side to the other. Well, the information so propagates through physical media in this case, but it's actually the information itself is decoupled from the physical chemistry of the media. It can be bits, it can be letters, words, and the media is not influenced about what is the content. But what happens in biochemistry? So cells have evolved to manipulate information in the biochemical environment. We know for a fact that one information molecule, very, very well known and important in life, is DNA. DNA can be also parsed into different kind of information. In fact, it can be very similar to a program. where we have different kind of subprograms, so parts of DNA that end up with a certain function. And we know that there is a certain information flow in biochemistry where DNA is transcribed into mRNA and is translated, not always, but it's mostly translated into proteins. Proteins are also information molecules, and we talked about that in these days. They are formed by a combination of a very long combination of 1 of 20 amino acids, and the way the amino acids are put together in a protein decides the relationship between different amino acids that are at each position in the chain, and that influences the way the attractions and repulsion of these amino acids are contributing to what is called the protein folding. So the protein is a sequence, but at a certain temperature, for example, room temperature, it starts having a shape because it's like a paper that, and we make a ball of that, and this is kind of an autonomous shape forming, and that shape decides the function of the protein. The protein can be, for example, helping metabolism, catalyzing other molecules, can be a building block in the cell. And this is a clear way we can observe how information here is not only abstract, but is actually structural. And this is one of the differences that I find between information in biochemistry and information in organic system. Obviously, information is also communicated between organisms, and the simplest organism, or let's say the building block of organisms, if they are multicellular, is a cell. And so this is where part of a molecular communication happens, where one cell communicates to another a certain state, or they exchange metabolites for higher level functions. And so what we say here is information is somehow coupled with the physical and chemical structure and the function of the molecules. And in the protein, we see that very well, but also in other molecules, in other kind of environments. So molecules and information, that's the very essence of molecular communication. Our reality is made out of molecules. Our reality is indeed a mixture of substances. A substance is one part of reality that is formed by the same type of molecules. And if we isolate a molecule, we obtain probably the atomic part, or let's say the smallest possible information unit in molecular communication. In fact, a molecule is the smallest identifiable unit of a substance and retains the substance identity and one important property, that is how that substance take part in chemical reaction. In fact, to have a chemical reaction, we just need one or a bunch of molecules or a countable number of molecules of that substance to observe a chemical reaction going on with other, obviously, molecules. So if we look into the characteristics of this kind of information, it actually lies in at the nanoscale, because a molecule is nanoscale big, and because of the strong forces of chemical bonds, I cannot directly, or at least with the tools we have now, access this information if I am not using chemical reactions. So the chemical reactions are really our way of operating and manipulating information in the molecular scale. Chemical reaction that rearranges the composition of molecules, and they are the primary processes when we think about molecular communication, transmission, and reception. And we talked about chemical reaction networks in the past days. Can be contained in nanoscale dimensions, and that's why molecular communication is also an inherently nano-communication paradigm. Okay, so having said that, if we want to summarize molecular communication field in a nutshell, We have molecules, we have chemical reactions, and last but not least, we have the transport, so the propagation of information, molecule transport. Many different ways of transporting information have been analyzed in the molecular communication field. The primary one is the Brownian motion, which somehow takes the role of the thermal noise that we have in communication, but also drives, for example, free diffusion as a process. All this can be considered if we look at the presence of a source of information, can be considered can be parsed into a transmitter process, a channel process, and a receiver process between a source and a destination. And that's what we call molecular communication link. Now, what is then molecular communication? It's the application of communication theory to molecules and chemical reaction. And that's how we define molecular communication from the very beginning, to characterize, classify, and predict, and model and engineer information flow at the molecular scale. So any molecular communication system can be kind of reconduced to this. And this encompasses both communication as propagation of information, but also communication intended as processes to store and retrieve information. So some of my past research, this is just some overview. We studied theoretically diffusion-based molecular communication, and we were among the first to have the first communication models of diffusion-based communication, molecular information flow for drug delivery. These are relatively old papers, and this is a subject of research that I recently got back to, thanks to a project. Then characterization of cell metabolism, using molecular communication, implantable device communication, even using neurons. So we looked at also propagation through neurons and neuronal cells. Design and engineering of components using synthetic biology, and analysis and redesign of signal transduction pathways, which are a way in biochemistry where information and also energy is transferred from a molecule to another. Anyway, if we look at molecular communication evolution, and we look at the human body, that is something that we some of the subject of our discussions in these days, we can observe that molecular communication, and in general, the field of communication applied to biological system is nothing but a multi-layer world. In fact, molecular communication can be found in system, organ, and tissue scale in the body. Just think about the cardiovascular system, and some of you have actually talked about that. Communication can happen also between cells and different kinds of cells. That can be cells that are part of our body and they are our cells containing our DNA, or they are as well part of our body, but they are of different types. For example, our gut microbiota. And also molecular communication happens inside a cell. There is transfer and manipulation of information inside each single cell of ours. At the molecular scale, we can access tools through synthetic biology to actually modify how these systems behave. And one primary way is to access the genes and modify DNA functionality. These, coupled with the possibility of interfacing to the outside world, for example, with legacy devices like electronics, gives rise to what we call the Internet of Bio-Nano Things, which is a concept where we interconnect both biologically based device, either engineered or natural, with legacy equipment that is electronics, for example, in implantable devices or wearable devices. And then finally, we go outside to the internet. So at the bottom of life, as I said, there are cells. And inside cells, there are complex chains of chemical reactions. They go on stemming from signals that can come from the outside environment. Now there is my picture there, but there are signals incoming. There is a clear information flow that goes from the outside of the cell till influencing how DNA is transcribed and translated. So the programs inside DNA are read and executed, many of them through the synthesis of proteins, proteins that can, for example, take part in metabolic processes, which convert energy from certain molecules, for example, glucose, into life of the cell, for example, biomass. So a cell can grow, or any other process that undergoes inside the cell. Synthetic biology is a way to tap into these processes. And we view information as a unifying quantity for biochemical process characterization. This is, in my opinion, where molecular communication is going. So we are interpreting communication systems in the biochemical environment, but the important essence is information. And this is a way to kind of give back to life sciences. We are providing a way that is quite unifying and multi-scale to view these processes, which is through the measurement and manipulation of information. So, stemming from this concept, I had the pleasure to work on a National Science Foundation grant where we were looking into how cells exchange information and how we can engineer through synthetic biology this system. And so within this grant, we also had the pleasure of having wet lab work, so some experimental work. We look into the idea of applying synthetic biology to design and engineer transmission and reception and obviously propagation of information, molecular communication links. And so some work stemmed from that. And what we did primarily was, first of all, understanding what is the limit in the exchange of information between engineered cells. And then how we can re-engineer part of these cells to include and enhance this communication and going towards addressing the limit. And so, for example, we studied how parity check coding could be program using synthetic biology in biological cells. And so these are some of the representative papers. This Infocom paper and this transactional communication are probably summarizing very well this work that we did in the past years. And then we looked at the top of life. At the top of life, we said we have the human body, which is probably the most complex which is the most complex life form around. And if we consider that we can engineer natural cells that are inside the body, so for many different reasons, for example, we might have artificial organs, tissues, and tissue regeneration, or even a better gut microbiome for our digestion. And if we consider the possibility that we can interconnect these engineered systems with legacy devices through different interfaces, we realize what we call the Internet of Bio Nano Things. And many of you are actually working on this topic, which is wonderful. And so in this respect, I had the pleasure to work on a grant where we were studying really how to realize these interfaces between engineered biological devices and electronics. And this interface, in the way we view that, was based on processes that in biology where in biology and biochemistry there is an exchange of electrons between molecules. And also there can be an exchange of electrons between molecules and an electrode. And so we envision this type of biohybrid electronics as in this case, a matrix of devices. Each single device is an interface. On this side, you have a biological specimen that can be a culture of cells or a tissue, and on the other side, you have electrodes that you can connect to circuits that can read out or can transmit information. And so this is a two-way interface that can also realize memory and computation at the same time through different processes. And so we had the pleasure to work on this grant. Another thing that happened through the years was the organization of a National Science Foundation, US National Science Foundation workshop that we entitled Biotic, Biology to Information Communication and Coding Theory. We were fortunate enough to be able to have this workshop in person in 2020 because it was just a couple of months before the world closed down because of the pandemic. And so we invited experts from all over the world to talk about how to apply how information theory is currently applied to biology and what are the future avenues for that. And so from these stems the work that I want to talk to you about today, because through this workshop, I had the pleasure to get connected to some collaborators. One of them actually is a mathematician from Case Western University who were trying to address the same question I was trying to address. And so we found a common ground and we started working together. This is thanks to a meeting, right? Like the meeting that we are having now. So I want to stress the importance of getting together in person and talk about research. So thanks, Christian, for this. Okay, so motivation. So again, Internet of Bio Things, we talked about the definition, but we didn't talk about what are the questions that we are trying to address here. So we need to understand how living systems process and intend information. And so the very start of this work is to understand how living systems intend information. And we already said that there are some differences. And also, there is a limitation because if we look at Shannon theory, we could measure information, but it's purely syntactic, does not take into account the processes of the living system, and appears insufficient. And in fact, we all agree, even in the title of this workshop, that we need a post-Shannon theory to be built. And so, how is information in biological systems? Well, organisms use information to navigate environments. So think about how information can affect survival and ultimately reproduction and the perpetuation of a certain species. So if you look at the classical models of information and you take into account a very peculiar example, all bits, which is all information, should be treated equally. Imagine you have a tiger. The tiger can be thirsty or hungry and can visually see a pond, for example, with water, or can see the gazelle passing by. And so there is an olfactory signal and there is a visual signal in this case. And so the value of information from the point of view of classical Shannon theory is exactly the same. And so in this case, the tiger would be able to choose between between going to drink or running after the gazelle only on the basis of how much information it receives. But that's not what happens. So information indeed has a semantic value, and so a meaning that is attached to that. And so information has a meaning and relevance. And so the information about the water and information about the food, the gazelle, is actually different. It has a different meaning. And there is also a fitness that is linked to that. So the tiger can say, I am more concerned about this information or the other, or it has a meaning. I know that if I look at the pond, that will bring me to drinking, and I know that if I run after the gazelle, I might eventually get meat to eat. And so there is a meaning that is attached. And this helps the tiger make the decision. And the tiger must decide what to do given the information that is available. Okay, but we agreed early on in our work that this is just part of the picture. There is another aspect of this information that goes beyond the semantics, which is the subjectivity. Information whose value depends on the context of the organism, which is a value that is very fluid. It changes continuously. And this is connected to the fact that the tiger at any given moment might be thirstier or hungrier. And it might take different choices, even if presented with the same information from the water and from the gazelle, from the ice. So, and it prioritizes sensing based on an internal need. And so, again, the same example, but now we attach not only a meaning to information, but also a need. So, for example, there is a higher need of water with respect to meat. And so, in this case, the information not only has the meaning of leading to drinking, but also has a certain value at that moment for the tiger, and this value can change anytime. So the tiger must not only decide which signal is more important, but actually also how much effort should we put into sensing. So what we are saying is that the very essence of the need of the tiger to drink or to eat will actually naturally put more resources, which means more attention to what the tiger sees. I put more attention to find a water reservoir than at looking at the gazelle. And we wanted to demonstrate that this is a guiding principle starting from very simple organisms. And so what we did is we developed a very simple model. It's a computational model. It's an automaton model of an organism. And we had a computational simulation of that. And we wanted to see the subjectivity as an emerging characteristics of how this organism uses and interprets information. And so this organism is a simple, we can think of a very simple unicellular organism. It has it's a motile species. The environment is monodimensional, which means this organism can only go left or right. And then there are two nutrients in the environment that are essential. So the organism needs A and B. It's like the tiger needs water and meat. So A, B, and distributed at different concentration around the environment, and the organism can absorb A and B depending on the concentration in the location where the organism is. So if there is more A and less B in that location, it will absorb more A and less B. And inside the organism, and the organism can divide only when inside both the organism there are two stores of A and B, and none of them has to run dry. If one runs dry, then the organism is perishing, it dies. But if the both stored information are over a certain threshold, the organism can divide and reproduce. The organism has receptors that can sense only A or only B, but it has both the two receptors in general, so it can sense both A and B, and senses a gradient. So it's divided into left and right, and can actually make a difference between what it senses on the left and on the right, and so have an idea of the gradient in that specific location of A or B. And it moves proportionally to the steeper gradients. So the organism follows what is the gradient that is the steepest, and it moves accordingly. And it constantly consumes store A and B. And if A or B are 0 at any given moment, the organism dies. So this whole thing, this whole model is to observe subjective information. And so two possible strategies for this organism. So given a total number of receptors and at any given time, the organism at all times can only use a certain number of receptors, but it can allocate the receptor equally between sensing A or B, which means that it gives the same importance to the gradient of A and the gradient of B. It's like the tiger gives the same importance to the water and the meat. Or it can adaptively allocate them. So this is an attention-driven mechanism. So if I have less A, means that I am more careful in sensing A. And so I put more receptors there, I have less noise in my measurements. Obviously, the more receptors, the less noise in sensing the gradient, the less uncertainty in finding my food of that particular type. If I need more B, instead I address that with more receptors of type B. The number of receptors stays constant, the type of receptor changes. We measured two things during this simulation. Information efficiency, which is the average mutual information at a certain time of all cells in the environment, and we have many at the same time. And we measured that as the average on the total number of cells of the mutual information that a cell is experiencing. Mutual information, which is obviously a mix of all the information that I receive from all the sensors. So I have the information that I sense on the right. This is the right side sensor for A. This is the right sensor for B, and the same for the left. And then growth rate. So the growth rate intended as exponential growth rate, so doubling rate, which gives us the idea of how fast the organism is actually growing, reproducing and growing. So how many times, or what is the tendency of the two reservoirs to go over the threshold and letting the organism reproduce. We distributed our A and B nutrients following a distribution that is called von Mises. So the von Mises distribution is a particular distribution that is the maximum entropy distribution in a monodimensional closed environment. Closed meaning that if I go, this is my range of motion between 0 and 100 locations. And if I go to location 100 over, I start from this side again. So it's just a closed environment. And the von Mises is the maximum entropy distribution within a closed environment. But we created two modes, one for A and one for B. So it's a multimodal distribution where A and B are maximally separated within this periodic environment. And here we observe some allocation of receptors, receptor A and receptor B, and we see that the divide density and the cell density at any location. Okay, so this is for the equal receptor allocation, and this is for the adaptive. And these are in times of simulation time, the location of, for example, two different organisms that we are following. Okay, and each cross is a divide event for this organism. And this is just to explain how the information goes. Important results. So we found out we have a parameter that is called cell stress, which is practically how much of these quantities the cell is eating for life, right? So every time step, the cell eats some of these quantities. That's why one of the quantities can happen that it goes to 0. And so as we increase the cell stress, we see that the information used by the adaptive receptor strategy is actually less than the one used by the equal receptor strategy. What does that mean? It's actually surprising. So it means that if the organism is adaptive, the information that is used more efficiently. So with less information can actually live better. And this is the growth rate, and we see that the adaptive receptor strategy can actually grow better depending on the cell stress, but for all the cell stress that we consider, than the equal receptor strategy. So if we put all these results together, we see that the adaptive receptor allocation, so the one that in our opinion includes the concept of subjectivity, and we will explore this in the next couple of slides. The adaptive receptor allocation results in a higher growth rate and correspondingly lower average mutual information. So lower average information that is used, that is even received, than the equal receptor allocation strategy. And this is very similar to what we think about when we think about the tiger. So what happens is the attention mechanism of the organism for whatever is more important at any single moment. Do I care more for A? Do I care more for B? Depends on my internal reservoir. How high is A? How high is B? And obviously depends also on how fast I'm eating the two. That's a cell stress. And so if I care at each moment about one or the other, in general, I don't maximize the information that I get from the environment, which is knowing A or B. So if I focus my attention, I get less information, but at the same time, I live better. Why? Because focusing my information on my need means that I'm addressing my need better. But overall, I get less information, which is somehow surprising because when you look at the literature of information theory in biology, many researchers, they point at the tendency of biology of maximizing information given the resources. And this goes against that concept. Now, but this is not the full picture. It's just a proof that something is happening, but we haven't yet found a measure for it. Okay. What we proposed that time, but this is just partial results, is to measure the information as the variance on the mutual information over all the cells in the environment, and then taking the average of it. But it was, and I want to just go over this slide because that was a very preliminary thought. And then we changed it when we studied another model. But what did we learn so far? So maximizing information efficiency can result in a lower growth rate. Equal receptor allocation, lower growth rate. Evolution can enable a diversity of perception in a population, and that's probably what is actually happening. And this is a new dimension to understand how living systems intend information, consider information, and process information. And it can be a novel inspired model to design communication system, molecular communication system in biology. And also, it's a push to revise information theory, post-Shannon, to address life. And now we move to another model. So this is the same situation, but the model is a little bit more complex. So, and more general. So think about our previous organism as this system, and whatever is around the organism is the environment. So the environment contains signals, for example, the nutrients A and B. These signals are acquired through an information acquisition strategy, and then they are processed with a processing strategy that ends up with a decision-making process on where to move. left or right, right, in the previous example. And at the same time, the organism has an internal state that can influence, in case of adaptation of the sensing system, can influence the sensing system, so my attention mechanism. And so there are two or more distinct signals in the environment. The system's response considers the internal state, and the system internal state is influenced by the environmental state. Yes, because the organism is eating the substance A and B, and the rate at which it's eating the substance depends on how they are distributed in the environment, right? And the system sensing resources are also finite in this case. Like before, we said the number of receptors is fixed. Okay, and so finally, we decided the way we wanted to define subjective information. And so that's why I put trademark there, but it's just a joke, okay? We didn't patent anything. So the difference is in the useful information. And that's why we talked about useful information here. So useful information, that is the information that is effectively used by the organism for its life. And we want to see what is the useful information between the system that can adapt its sensing, that has this attention mechanism to its internal state, and a version of the same system that does not have adaptation, but looks only at where information is and what has the most information. And so we define subjective information as the information of a system that can adapt minus the information, useful information of a system that cannot adapt. And what is the useful information? It's the subset of all the information available to the system that is useful for the organism to take action and live and reproduce. How do we account for that? Is the expected value over the internal state of the mutual information. So it's the expected value over the internal state. A and B are the internal state, how much A or how much B I have, the two nutrients, and the action that the organism decides to take, moving left or right, average of this over all the organisms, and obviously depends on a strategy. A strategy is either to adapt or not adapt. So when we compute all this, we believe we can measure the subjectivity or the subjective information of that particular biological system. And we believe this is a new metric to characterize a biological system. So, yeah, this is two relevant systems, the system response. So what we observe with the previous model that I told you is that this is a further proof that we wanted to give, is that if I utilize these two sources A and B and I separate them with respect to not separating them, I have here that the syntactic information, which is the information without any connection to its meaning, right, does not really show that it's correlated to the growth as much as instead the subjective information, where if I have my two nutrients that are divided, that are separated in the environment, so here is the concentration of nutrient B, concentration of nutrient A, if they are separated with respect to if they are in the same location, obviously, I can see that there is a correlation between the subjective information in the way we measured it right now and the growth, right? But obviously, if they are co-located, I don't have any difference in the information between A and B because they have exactly the same gradient. Then I lose that subjective information importance, and I have just my blob without any shape. Instead here, if I make a PCA, which is what represents this line, I can see that there is a correlation. Okay, so what we did is we define and analyze a minimal model for subjective information emergence. And we looked at how an organism can gather information to maximize its fitness with the two strategy. And again, the organism is, again, unicellular, is capable of sensing environmental concentration of essential nutrients. And we consider these two, A and B, like before. And it's directing its movement accordingly through what is called chemotaxis, which is this gradient sensing and then decision whether to move left or right. But in this case, instead of just performing simulation and analyzing the results, we actually have a full analytical solution of this system for two different quantities. The expected incremental reward gained by the organism throughout its lifetime, which is, well, here is just expressing a average over all the lifetime of the organism of a reward, of an incremental reward, and also in terms of the useful information, which we computed using the previous metric, used to achieve that reward. Again, let me remind you, useful information means the amount of information that effectively the organism uses for living, okay, and for thriving, for surviving and reproducing. Okay, so what we did is we made a new model. In this way, the model is simple enough that we call it minimal model to achieve an analytical solution of the whole system. So we don't need simulation anymore. We just solve all the stochastic equations, getting to the result. And so now the environment is simplified to only 3 locations. I have different combination of the concentration of A and B, and I have different combination of how these are distributed in the environment. And the environment is modeled in this way. And also we have that the organism is very simple. So I still have receptors A and B. The total receptor count is always kept equal, but now the concentration of A and B can be only one of 3 values, 0.25, 0.50, and 0.75. And then we assume that the unicellular organism can have 3 possible location and environments, that A and B are essential nutrients again. And in this case, to achieve an analytical solution, we consider all possible permutation of the distribution of A and B in the environment. Remember, before I had a fixed distribution, there was the von Mises motivated by entropy arguments. In this case, instead, we consider all the possible combination of the nutrients. It's like taking the tiger and considering all possible combination of water reservoirs and gazelles around, okay? But in this case, it's just 3 environments, so it's much easier. Just two nutrients and just 3 possible combination of concentrations. The distribution can exist in 3-form uniform random environment. The cell can absorb, again, A and B, and what it absorbs become part of the internal reservoirs of A and B. The cell can sense the nutrient with the receptor, so ligand binding receptor process. The total number of receptors is kept constant, and there can be an adaptation in the number of receptors that are allocated for substance A or substance B. And then movement decisions to move left, right, or to stay in the same location, so -1, +1, or 0 in this monodimensional decision, monodimensional line, are based on the number of bound receptors and the internal state. And also I have the stress. So the organism is eating. And here I also put a stress on the total number of receptors. It is fixed for an organism, but if I were to change it and consider different number of receptors, I can consider that if I have more receptors, I spend more energy to synthesize them. Imagine they are proteins, for example, on the membrane of the cell. So I have to spend energy to allocate these receptors, I have to spend energy to synthesize these receptors and having them on my membrane and ready to sense something. And we consider that receptors for substance A and substance B, for simplicity, they take the same cost to be synthesized. And so we looked at the expected incremental reward that is based on the goal of the organism. So a 10 time step, and again, it's not a simulation, is an analytical solution. We maximize the lower of the two internal stores. That's what the organism wants to do, right? If I want more A, I want to maximize the way I catch A, or vice versa for B. And so the incremental reward, the reward for a strategy is the average incremental reward, and the incremental reward is given by the reward of the organism with respect to the number of allocated receptors, if I can change them, minus my stress. And so this is the internal state, which is the internal A minus internal B. And obviously, I want to have A and B as much as possible in equilibrium. This is an ergodic property of the internal state, and that's why we can solve it pretty easily. So in the adaptive strategy, I have that my reward is equal to this expression. This is the reward that take into account the maximization of the receptors A and B. And this is a probability term. And this term means the probability of having a certain internal state given the receptors that I have allocated. So these two type of receptors, A and B, they can be allocated out of a total number, as I said before. And this maximizes the expected reward per internal state. So the cell can allocate optimally these receptors to maximize the reward, which means to equilibrate as much as possible the quantity of A and B internal. And then, this is a stationary probability distribution. And then the non-adaptive strategy is exactly the same, but in this case, I don't have adaptation. So the number of receptors for A and B is just a half of the total receptor at each time. And so I have fixed allocation for A and B, and I have also stationary probability here. So what we did is we considered the reward per internal state, which means upon movement is the reward that the organism maximizes the reward over all possible numbers of bound receptors and their distribution depending on the strategy. So either I can allocate receptors for A and B, or I cannot, and so I have them fixed. So it's a stationary probability distribution, and we demonstrated that in the paper. The distribution of the internal state and receptor allocation over all possible location and environment, okay? And I have a total of 9 equiprobable positions. So long story short, what we did is we measured the subjective information and we measure the useful information to measure the subjective information, and the useful information is calculated as the expected value over the internal state of the mutual information between the concentration, distribution of A and B across the environment, and how the cell moves, right? We had in the expression before action. So here action is the movement, and it depends on the strategy in general and the internal state. Okay, so do I need more A or B? And I make a decision accordingly. So we obtain analytical solution for receptors with a dissociation constant of 2. We have this is the constant penalty term, so how much of this nutrient is the cell eating at each time step, and then the cost per synthesizing a receptor. And we also provide all code for analytical solutions. So if you want to try this out, we have everything in GitHub with all the code. And also my student Tyler is a very good programmer. So he also commented all the code very well. So you can follow exactly whatever we did in the paper. And so what we found out is that the cell here, what we can appreciate is that there is a relationship between the reward and how we allocate, or what is the optimal allocation for the receptors. And what it turns out is that if I can reallocate my receptor for A and B, it is the best strategy if I need, for example, more A, is to allocate temporarily all the receptors to be sensing A. And then if I need B, is to allocate all the receptors only temporarily to sense B and forget about everything else. And this is one first results from the whole analytical solution. So the organism's expected reward is dependent on how the organism at the end subjectively acquires information. And that's the next result. So this is the useful information with respect to the strategy. So either adaptive or the one equivalent, so that doesn't reallocate the receptors, and the incremental reward of the strategy. And so what we see here is this upper curve and this lower curve. So the lower curve, if you see, is related to an organism that cannot adapt its receptors. And the upper curve is related to the adaptive organism. And we see that the adaptive organism, and we see these 2 points, for example, this is for different number of receptors. These different points, they have a different number of receptors. And we see that for 100 receptors, for each one of these two curves, I obtain these 2 points. So if I make a comparison between these 2 points, I can see that there is a distance. So this distance between 2 points denotes 2 things. One thing is, well, the adaptive strategy has higher incremental reward, which means that the adaptive strategy makes the organism live better or longer. And the second thing is, on the x-axis, I have also a difference in useful information. What it means? It means that the adaptive strategy has more useful information than the strategy that it does not have adaptivity, which means that an organism that has a subjective approach to information lives better than the organism that is kind of neutral with respect to the information. Okay? And so this is very, very important and very striking. And so adaptive strategy can gather a higher amount of useful information that contributes to wiser choices for the organism's movement, and then in turn leading to a higher expected reward. So the organism decides, I want more A or I want more B, adapts its information acquisition apparatus, very simply here, just to receptors, and then ends up living better because their movements are better informed. And so it can actually look for A and B on the basis of the need in a better way. And this is a result that I really like. So this is the reward per bit of information with respect to, as function of, the receptor count, so how many receptors I have. So you will agree with me that if I have a sensing system in molecular communication, the lesser receptor, the more noise I have, right, in sensing, the more uncertainty I have in sensing a concentration. And so this result says that as the noise of the information acquisition increases, so I have less receptors, so I have more noisy measurement of the concentration of the nutrients, the fitness value of the subjective information increases, which means that the subjective information or having the subjective component is more and more important, the more noise I have in the system. So the more uncertainty I have in sensing, the worse are the eyes and the olfactory system of the tiger. The more important for the tiger is to adapt its attention to the need. So I'm more careful in spotting the water around, or I'm more careful in spotting the gazelle running. And this is the more important for more noisy measurement. So if my vision is less capable, or my olfactory system is less capable, it's even more important that I concentrate my attention to that particular apparatus on the basis of what I need at the moment. So we develop an analytical model to compare adaptive and equivalent strategy. We solve the model to demonstrate that adaptive strategy provide higher growth and reward by prioritizing subjective information. In the paper, we have the whole analytical result. It's very complex, and I hope I simplified that enough just for this talk. But the way we solve that analytically is actually a very quite a lot of equations and relationships. And what we did is we found that the difference in reward function effectively measures the impact of subjective information on system performance. And it highlights the importance of context-aware information processing in maximizing fitness, and defines a new measure of information, which we called subjective information. And we propose this subjective information of being one of the type of measurements that we should add to information theory in a post-Shannon era. So just a highlight of something I'm working on now. So we are simulating the evolution of molecular communication mechanisms. So we are actually using insights into biological processes to guide how molecular communication can be not only designed to work, but designed to be optimized. And so what we did is we use a Lewis game framework to simulate a molecular communication system where each node exchanges molecules through a diffusion channel, in this case, free diffusion. And what they do is that they don't have a fixed coding of information, but they adapt through a learning process. So it's a machine learning process among all these nodes. So it's a collective machine learning where I have the need to transmit this information X from this node 0 to this node 3, which is the recipient, which has to get to information Y. And to do that, the nodes have to self-adapt parameters in their encoding process, and they use a bunch of molecules to transmit this information. And so this is just an example of this system. We had many iterations where I have many nodes distributed in the environment, and each one is learning a coding. Each color is a different quantity of a certain molecule type. Here we had 4 different molecules types and six different nodes, and they are transmitting to each other. And these are some results of how the learning works. And I have number of nodes versus test accuracy. And we found out that in this case, the lesser node we have, the more accuracy we have for the system. But we are still studying. These are just some preliminary results on this. So the concept here is to let the system self-adapt its own molecular communication to be able to be maximally fit to the applications that we are trying to realize. Okay, so I want also to invite you, I know some of you will be already participating, to Catania. They are holding the 9th workshop in molecular communication. And I'll be participating because, well, I will present my research there, but also because we are organizing a panel in collaboration with the IEEE ComSoc Italy. And we will having a high-level panel with short lightning talks of 5-7 minutes, and they will include both academic researchers and industrial people. And we will have open discussion with student, faculty, and international collaborators. And it will be in the afternoon of the second day, so Thursday, April 10th. It's a free event, so anybody can join, even if you are not registered at the workshop. And it's all offered by IEEE ComSoc Italy. And if you are already part of the workshop, consider attending. I know Catania is beautiful, and probably you reserved that afternoon to visit it. But stay with us for some hours more before going away, and help us have this panel discussion. And if you can also present 5-7 minute lighting talk about your research, that would be great. Some of the talks that I've seen here, actually all the talks that I've seen here will be actually a great fit for this. Okay, so another invitation is ACM NanoCom 2025 that will be in Chengdu, China, this following fall, October 23rd, 25th. The call for paper is out, so consider submitting a paper. I'm part of the steering committee, so it's my duty to remind you about this. And also, I remind you that we have a journal. I am co-editor-in-chief with my esteemed colleague, Professor Jornet, Nanocommunication Network Xavier, open to all your papers and submissions. Okay, so these are all my thanks, and I have many more, and I couldn't fit on a slide. All my collaborators, students, including grad, undergrad, master, PhD, visiting scholars, and so on and so forth. And thank you so much for having me and for having this opportunity to talk to you and network with you in this very good workshop. Thank you.
Okay, yes, thank you a lot for the talk. And are there questions or comments?
Hello. Yeah, thank you very much for your talk. It was very, very interesting. I liked it a lot. And I have a couple of questions. So
Please.
I have not worked much in molecular communication. I'm here mostly from the post-Shannon point of view. And there is something very interesting that we're doing with some colleagues from TU Munich in post-Shannon, which is these new metrics like value of information and age of information that are not related to molecular, right? We have a very simple example with our colleague where she tries to balance multiple inverted pendulums that communicate with a controller. The thing is that if the resources are limited and you put the same value on every packet that they transmit, eventually the pendulums will fall. So she has this concept of value of information in which when the pendulums are falling or the packets are new as the packets, that's the one you have to prioritize in order to maintain them available. So my question is, have you considered using your metrics, such as subjective information and such, for these non-molecular communication approaches like value of information or age of information?
Okay, that's an interesting point. So it depends. So I think that the subjectivity is something that is connected to a concept of survival, so birth and death of an organism. And as long as we have a system, a framework where we have a birth and death, and probably your pendulum, when it goes down, is actually a death process, right? Yes, we could, in principle, try to apply subjectivity there too. And again, subjectivity is different than semantics, because semantics is the meaning and subjectivity is the interest on the meaning somehow. We studied something related to the age of information as well, but your suggestion, I think it makes a lot of sense, and I want to study more before having a definitive answer. What I can tell you is that this latest work that I presented is now being extended to be incorporated in a framework that considers rate distortion theory. So we found out that we can, in principle, find a rate distortion curve where we see the effect of the subjective information. And I couldn't present it here. It is really fresh out of my PhD student studies. And it's being now written into the journal version of this paper, which we will submit very soon, and we haven't yet submitted. But that is the extension that we are doing. But what you are considering is to get even more general and consider systems that not necessarily are living, but they have a process. And in principle, yes, in principle, I can tell you it makes sense, right? As long as you have a system that has this, that you can reconduce to this same framework, then yes, you can study subjectivity and subjective information also in that case. And probably if your system can be modeled with a concept like this, then yes.
Yeah, I think that would be a good connection because, as you mentioned, right, the pendulum falling down could be related.
And I'm willing to talk more about that. Yeah. Thank you so much.
I have a couple more questions.
Please.
If we have the time, right? So you mentioned that current information theorists, when they approach this concept of living organisms, they tend to assume that living organisms maximize the information given their resources, right? I have no clue about this idea or this topic, right? But for me, that was a little bit shocking because what I know about living organism was what Pitt was doing. And actually his brain cells there in Australia, they try to minimize the entropy because that's how you punish them. You punish them with high entropy signals, and then you reward them with low entropy signals. And apparently brain cells crave these low entropy signals. So it's very interesting for me to hear that information theorists were taking the other approach in their models. So what are your thoughts on this related to, for example, Pitt's talk on?
I mean, this is evidence. So it actually just feeds into this whole concept. And I completely understand your surprise to know that information theorists in biology have always pursued this maximum information concept. But if you look at the literature, and it's really, so it's almost a separated community with respect to communication engineer. Those are really like mathematicians that are looking into evolutionary processes as how information plays a role there. I can point you to some papers. And there was one paper that was trying to demonstrate that an organism, if you give an organism a certain number of resources, it didn't really demonstrate, in my opinion, but it just says that the evidence shows that the organism will always be willing, or it's always better, let's put it in these terms, it's always better for an organism to know the most about the environment than to know just a portion of it. So for example, if I have eyes, it's always better that my eyes are seeing every difference in the environment and every contrast with respect to just seeing one portion of the information that is in front of me. And this was going to the principle that, well, no matter what the organism does with the information, it's always better to know more. And then what we contrasted was this concept where, no, if I know more, sometimes that knowing more is added information that not only is not useful, but it's also detrimental because it can influence my choices, not in the way that I would like to. And so it makes more sense to say I focus my attention on something, and then I use that information. That is my use. So I maximize the useful information for my survival. And that's what our intuition, then we did the simulation and we found out those differences. But then finally, we had this analytical proof. And I would say that with this analytical proof, we were very satisfied. And for now, it's just on a conference paper because we just wanted to get it out as soon as possible. But now we are transferring it into a journal paper. But both the framework we are looking into the ray distortion theory framework, but probably also your concept, and now I want to explore it more, of like saying, well, yeah, we have an organism, but it might be also something else that make a lot of sense.
And
Yes.
my last question, so it's a very interesting model, right? So it's very nice, a very simple toy example that could lead to further development there, right? I think the killer would be if you could think of a real life experiment that could validate this. Have you looked into this? Have maybe with medicine people, with bacteria, with biologists, that can allow you to validate this simple model with simple organisms?
Okay, so yes, it's been months. We are brainstorming together. I don't know if you ever heard about Domitilla Del Vecchio in MIT. So she's an MIT faculty, and she studies systems and control in synthetic biology. And so we contacted her. I knew her already for a long time, and we also had student exchanges. And we wanted to come up with an organism that is exactly doing this, where we can observe. And we have been debating for a long time whether we should use a natural organism. So we say, oh, we took it from nature, so it's actually how nature works through evolution, or a synthetically engineered organism. Obviously, if you synthetically engineer the organism to behave like this, is easier, but at the same time, we don't want the experiment then to say, oh yeah, if I engineer it, it works like I designed, but the natural organisms, well, we are not observing them, and so we cannot claim that through evolution they came to this. And so through her, we reached a microbiologist who knows a lot about wild type organisms. And we asked for a suggestion for an organism. And the answer is it's very complex because it's difficult to isolate processes like this on a very dense network of interrelated processes. And so practically the complexity goes up exponentially, and our toy model is not, it becomes like a hyper complex model. And so we haven't found yet a good organism for that. But I can assure you that the microbiology expert is one of the worldwide renowned experts that we contacted, and he was skeptical on the fact that we can find an organism to really isolate these and make it like similar to the toy model without adding so much complexity, then it's not valuable anymore. And so where we are going now together with this MIT faculty, Domitilla Del Vecchio, is to the synthetic biology route. So we will engineer an organism to do things similar to the toy model. At the same time, we'll try to engineer the list. So having a very low intervention. And so we are exploring different gene knockouts now and things like that. So that's what you're doing. But yeah, I mean, that's the natural advancement of this work is to go and do experiments and then see. We also observe that in real life. Yeah, that's what we were trying to do. Yeah.
Thank you very much.
Right.
Thanks again for your talk. It was very interesting.
Thank you.
Yeah, well, I don't really know what question I want to ask, really. It was a wonderful presentation. Thank you very much.
Thank you so much.
So I was thinking precisely on this, maybe a natural application for this, and thinking about how cells really behave or how this would scale in a natural model. And I was thinking that, well, on how natural it is to make this energy spend that you're calculating, this energy. Well, you spend these resources to move smartly, let's say, inside your compound or your solution. What if, or have you thought about, instead of spending the energy moving smartly, you spend the energy on processing the information? I mean, just having the cell move randomly and just processing information in a clever manner makes you spend the energy. If you process a whole sequence of a protein, let's say, you spend more energy than if you just process and discard. Okay, this is not a cell, this is not water, so I discard it.
It's a great point. So we haven't associated energy to processing right now. It's natural to do that because natural systems, might spend more or less energy in processing. Just think about how the cells process. As I showed at the beginning, we have these dense chemical reaction networks, and obviously, the more complex processes I need to do, the more complex is the chemical reaction network, the more energy it will consume. So it's natural to associate a energy consumption and a penalty to the processing. We haven't done it because we wanted to just really isolate very simple processes here, and we wanted to give a value to this attention mechanism. But one extension of this is exactly what you're doing, where we look at the processing. Now, you mentioned this random movement, and it's very interesting. I haven't thought about it. So moving randomly, it's one possible. So one possible other system that we could study is that one. I mean, the limit is the sky here, because you can come up with a lot of different thought experiments to reveal this subjectivity, right? And the one that you are mentioning, where the processing takes the place practically of the movement, is actually very interesting. I think it's something we can explore. Yeah.
That's fantastic.
Thank you so much for this.
Thank you.
Yeah, I actually will note it down, so I don't want to forget.